the muscle anatomy. And it won't take us a tremendous amount of time next week to do that. I don't see it taking the full lecture time. So I told you guys to go ahead and print those out for yourself if you want them for today. For lab, it can help you. Today's really chapter 11. And this is a chapter that for some can be a little complicated just because it's dealing with physiology or mechanisms of action that sometimes are hard to visualize. And so we'll try to do a little bit of that here. In fact, I might even go get them out. Yeah? I have a couple. Anybody else need a copy of 11? That's all I got. Okay. So today, as I said, is basically going to deal with how muscles work. And you'll have to go back to chapter 3 a little bit in your head, and we'll talk more about that with the sodium-potassium pump. Remember that idea. The resting membrane potential, that's where the membrane, the plasma membrane is slightly negative on the inside. We'll come back to that idea as well. So I told you with chapter 3 that would come back. Here we go. This is the first of 2 lectures we'll deal with that specifically. So let's go ahead and start chapter 11. I'll give you a warning right now. I don't think I'm going to finish by 7:30. The lab will be short to make up for it. So don't fret if we go to 8 or so. I'll give you a break, or maybe even 2 if we need to, but I don't think we will. Okay, so just basic review. We talked about there's 3 types of muscles. We have skeletal, cardiac, and smooth, something we talked about before. There's several terms here that you're going to see mixed throughout these lecture notes. In general, things like MYO or myo, things like MYS or sarco, all refer to muscle. So you're going to see a lot of the terms have these in there. So for example, a sarcolemma. Sarcolemma is actually the plasma membrane of a muscle cell. Okay? Sarcoplasm is actually the cytoplasm of a muscle cell. And then sarcoplasmic reticulum, or SR. This is a very specialized type of smooth endoplasmic reticulum. Very specialized for muscles. And you're going to see how that is in just a little bit. So again, you're going to see more of these terms later on. I just want to give you the heads up with those terms right now. So, compare and contrast skeletal, cardiac, and smooth. That just shows you right up here. You can see the differences basically are underlined for you. Real simple. Skeletal muscle is, well, found in the skeleton, right? This is where we have our movements on the skeleton. They form levers. We talked about that last week. So these are our voluntary movements. We voluntarily move our arms, move our legs, move our mouths, etc. It's also striated. So here's a picture to show you the striation. You've probably seen this once before, but these are the striations. You can see this striated appearance. See what I mean? The little ridges. You see the striated appearance on skeletal muscle. And as I said, it's also voluntary. So we have skeletal, striated, and voluntary. Cardiac muscle, we're not going to really talk too much about in this class other than really right now. You'll talk about that more in 102. So cardiac, as you remember, is also striated. It has intercalated discs as well. We're not going to go into that now. Cardiac muscle is only found in the heart. It's the only place you find it. And it's involuntary. We can't control it voluntarily. Our heartbeat, for example. Now we can have an emotional response to it that'll change it, but we can't consciously control that. And then smooth muscle. Smooth muscle is smooth. There are no striations. So it's non-striated. It's also visceral. What that means is the visceral tissues or the visceral muscle, for example, around your intestines, that help squeeze the foodstuffs through the lumen of the intestine. That would be considered visceral. And I said it's also involuntary. We can't control that. It's not something we can voluntarily control. Simple things we talked about a little bit before. Functional characteristics of muscle tissue, again, not something I would specifically ask you to list the 5 or so features. I think that's just something you can read on your own and understand. But responsiveness, or what we call excitability. So we can respond to stimuli. Muscles will respond to a neurotransmitter. And we'll talk about acetylcholine. It's one of those neurotransmitters today. And in so doing, they respond by contracting. So they're responsive to outside stimuli. Conductive, or conductivity. That means we're going to see a local electrical charge being changed along the plasma membrane, or what we call the sarcolemma. So I'll explain in 2 seconds. Won't expect you to understand all of it right now. But if you remember the resting membrane potential is negative 70 millivolts. You're going to see that change really quickly in one area of the plasma membrane. We're going from negative 70 up to about positive 30. That rapid change is a voltage. And you're going to see that propagate or travel down the entire length of the cell. Okay. Come back to that in much more detail later on. But that's the conductivity aspect. Contractility. Muscles shorten. People think that muscles will pull. That's true. They don't push though. Okay? Muscles pull, they don't push. So we'll talk about contractility. In other words, shortening. Muscles shorten, and that's how they do work. Extensibility. Muscles can be stretched, and we'll talk about how muscles can be stretched, but that's not how they do the work. Okay? And then elastic, meaning they have some ability to recoil back to their original position. They actually have little rubber bands inside of them, for lack of a better word, that basically brings them back to their original shape. Okay. You can read the muscle functions on your own, skeletal muscles. Let's talk about, let's go to here, this big picture here. Here, I'll start here. This is actually something you'll see in chapter 10, not something you'll see in chapter 11, but I wanted to show you this real quickly so you can see the big muscle. So this is a muscle right here. This is one representative picture of a muscle. I mean, this is a giant muscle right here. And next week we're going to talk about these different membranes that wrap around the muscle itself, that wrap around these little bundles of sticks, and even wrap around individual muscle fibers. We're not talking about that today, but I wanted to show you this picture so you can really understand what we're talking about. We're going to spend 90% of our time today talking about this right here. This is a muscle cell, or what we call a muscle fiber. It was once many, many cells that then became one giant cell, or it's multinucleated. And it exists as a long tube-like structure, so they call it a muscle fiber. So whenever I use the term muscle fiber, I mean muscle cell. I know, it used to confuse you, right? A muscle fiber, or a muscle cell, is packaged in this little bundle of sticks called a fascicle. And then fascicles are arranged in different ways to make up a muscle. And again, we'll talk about the different arrangements of fascicles for those bundles of sticks next week. So not something we're being concerned about right now. I just show you this picture so you understand we're talking about that little detail. One muscle cell, or muscle fiber, in the entire muscle. We're going to spend all our time talking about that.
Is this one of those instances where, you know, when infants are born, it's one nucleus per muscle cell, and then as we grow older, it all, no?
No, it does that, well, it does that in utero as we're developing. Yeah. So this is a muscle cell, or muscle fiber. Okay, so this is that big, big picture, now with that little small detail I showed you before. So this is the plasma membrane of the cell, what we call the sarcolemma. Come back to that term several times. Notice that this cell is jam-packed full of these big collections called myofibrils. And those myofibrils contain these little myofilaments we'll talk about later on, or the thick and thin filaments. So we have these filaments here, thick and thin, that are bundled together in myofibrils, and then the entire cell is jam-packed full of those. You see that? Remember I said the sarcoplasmic reticulum is a specialized structure? When we talked about it in chapter 3, I said different types of cells have different types of smooth ER. This is one of those examples. So the smooth ER of a muscle cell is highly specialized. It has been designed to wrap around these myofibrils, and you can see that here in blue. So you can see a nice little network, or almost spider web-like network that wraps around these myofibrils. And then you'll see these large tube-like structures here and here that are flanking these purple tubes. And we're going to talk about those purple tubes in a few minutes. Okay. The blue network, or the sarcoplasmic reticulum, exists to have one main function. It sequesters or holds onto the calcium. So it holds calcium. It's a reservoir for calcium. So the calcium levels in the cell, the muscle cell that is, are kept fairly low. Until the sarcoplasmic reticulum is triggered, at which point it releases calcium into the cytoplasm. And that leads to contraction. And that's what we're going to talk about, that whole process. So how does it get triggered? How does it get turned on? In general, we're going to talk about that electrical impulse coming down the plasma membrane. We're going to talk about that change. You can't see this very well in this picture, but these purple tubes are basically continuations of the plasma membrane. So an electrical impulse that will come down the plasma membrane of the sarcolemma will then travel down these purple tubes. Travel down the purple tubes. Travel down the purple tubes. We call transverse tubules or T-tubules. Transverse tubules or T-tubules. So as that electrical impulse comes down, it'll cause the sarcoplasmic reticulum to release calcium. That's where we're going to go in more detail. Okay? So I don't expect you to understand it in my 10 seconds of explanation. You're going to see a lot more. But that's where we're going to go. Okay? So we're going to talk about how the electrical impulse gets generated. We're going to talk about what happens to calcium, why that leads to contraction. Okay? That's where we're going. A couple other things here. We'll come back to this idea of glycogen. So if you look at the top of page 3, it says granules of stored glycogen. Glucose. We're going to use glucose to build or make ATP, regenerate ATP. Remember our charged batteries? We need energy to cause muscles to contract. We get that from glucose. So we're going to have a polymer of glucose called glycogen stored in our muscles. So that if we need a burst of energy, we can build ATP from that glucose. Make sense? Stored glycogen, that's why we have it there. Myoglobin, just something else to be familiar with. Myoglobin makes the muscles red. It's the oxygen-containing protein that is found in muscle cells. It's similar in composition to hemoglobin, which you find in blood. So I always laugh when people, break out a steak and they say, "Oh, this steak's really bloody." And I always say, "No, it's not. It's just got a bunch of myoglobin in it. It's not hemoglobin. It's not blood." My other class got Okay, so myoglobin is the oxygen-containing component. It's really what makes meat red. Okay? It's not really blood per se. Okay. Let's look at this picture here. We're going to go to this picture and then this one all together. Okay, so let's just focus on this one first. This is the kind of picture you see in your exam, so you know. We'll come back and start with this one first. So we talked about the striations. The striations, or that banding pattern of light, dark, light, dark, light, dark that you see in skeletal muscles. Okay, you also see in cardiac, but right now we're going to be talking about skeletal for almost the entire lecture. So here in skeletal muscles, you see this banding pattern. And there's a name for these bands. So the light bands are called I bands, and the dark bands are called A bands. And so you see alterations of A to I, A to I, A to I. Yes, you should know that. Okay. Within each I band, or the light band, you'll see this dark line. We call that the Z disc. Z disc. Within the A band, or the dark band, you'll see a lighter region right here called the H band, and then right down the middle there's called an M line. You'll see that in a few minutes. It's not really labeled there. So H band and M line. This right here, we're going to come back to over and over and over again. The distance between this Z disc and this Z disc right here is called a sarcomere. And it's the smallest contractile unit of a muscle. And you're going to see why as we go on. Let's move on to this picture here, and I'll show you the same thing we just looked at. You give me your quiz. You what? Okay, you just want to not turn it in.
What's your lesson for numbs and all? That means
Well,
two quizzes behind.
Okay, well, let's talk early break.
Yeah, I was going to tell you.
That's fine. Okay, so this is the diagram or the artistic impression of what we just looked at from the microscopic picture. So this here is the A band. That's that dark region. This here is the I band. And the reason why it's light, or the reason why it's dark, is because of the fibers, or what we call the filaments, that are inside it. So we talked about thick and thin filaments. We're going to talk about that in much more detail. Here is a thick filament in purple. A thick filament. The thin filament is right here in red. And the green we're going to ignore. Okay. I'm not going to talk about that. It's just elastic filament. It just makes muscles kind of spring back on themselves. Nothing I want you to concern yourself with. We're going to be concerned with the thick and the thin filaments. And so you can see right here in the middle, the reason why it's so dark, the reason why it looks like a dark band, is because it's got the thick and the thin filaments on top of each other. And that makes the overall effect very, very dark. Except for here in the middle, where it only has the thick and not the thin. That's why it's slightly lighter there, what we call the H region, or the H band. Then there's a nice little protein we call the M line right down the middle. That's where that comes from. Over here, you can see the I bands here and here. And the reason why they're lighter is because there's no dark or thick filament there. All we have is the thin. And then right down the middle, we have the Z disc. Does that make sense? And again, the distance from here to here, Z disc to Z disc, is called a sarcomere. Sarcomere. And it's the smallest contractile unit of a muscle. Underline that, star it, do something important with that one, because that's going to come back. We'll talk about that more. Okay. So you can see in this picture that this thick filament right here, or microfilament, is right next to the red. And what you're actually going to see in just a little bit is it's literally going to grab onto the red and pull. And I wish I had a ruler. I had one in the other class. I should go find one. But you can imagine, I'm that dark fiber right here. I'm the thick filament. And I'm going to grab the red and I'm going to pull and pull and pull. And in so doing, I'm going to bring the red filament closer to the middle. Okay? Same thing over here. We're going to bring this closer. So we're going to pull. This guy's going to pull this rope this way. This guy's going to pull this rope this way. And what you're going to see, okay, is the distance from this Z disc to this Z disc is going to get shorter. That's where we're going with this. Okay? Let's look. Here, let me show you. I'll just show you the picture because I have the picture here. It looks like these guys are just one-to-one ratios. We have one thick and one thin, and then a thin and a thick and a thin and a thick. In reality, this is the true three-dimensional structure. Okay? It's kind of above and beyond what we're going to get to in this class, but in case you're curious about it, there's really 6 thin filaments for every one thick. And that's how the arrangement really is in a muscle cell. But for understanding the physiology, a two-dimensional picture is sufficient. Okay? So this is the picture, as I said, I would show you on an exam, for a lab exam, for example, would have you label it appropriately. I say, name that band, et cetera, et cetera. So just be aware of that for the upcoming lab exam. Let's talk about the thick and thin filaments. So this is a bigger picture of the thick filament. And you can see that what it really is a bunch of myosin molecules all packed together, all oriented in the same way. So all their heads are this orientation on this side, and over here they're going to be the opposite orientation. You'll see what I mean. So the heads are right here. These heads, this is the best way I can describe it, it's like my hand, okay? What I'm going to do later on, I'm going to show you how the head gets cocked with energy in a cocked position, okay? And in this position, I can grab the rope and pull. Cock it, pull. Now imagine tug of rope, right? Tug of war. And I'm cocking my hand and pulling the rope. Cocking, pulling. And what's going to happen? That rope's going to get closer and closer and closer and closer. So what you have here is a bunch of hands grabbing that rope. Pulling, 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 pulling. Does that make sense? So this, on this side is pulling this way, and this on this side is pulling this way. So go back to this orientation. Go back to this picture. I'll show you what I mean. These guys here are pulling this red rope this way. Pull, pull. And these guys over here are pulling the red rope this way. And if you're pulling them this way, what's going to happen? The distance between this and this are going to get closer together. There are probably videos and all kinds of things online if you're not visualizing this better. but I just don't have time to show you that today, okay? So this is the thick filaments, as I said, it's made of myosin. Yes, you should know that. Myosin is what the thick filaments are made of. Thin is made of what we call actin. Actin. This is what thin looks like. This is a thin filament. Now you can't see it in this picture here, but later on you can see a much better picture. There is an active site being blocked right here. There's a little hole right here. And all myosin, all the thick filament wants to do is grab onto that active site. It's sitting right here and it's waiting to grab onto it, but it's got a problem. This molecule called tropomyosin is blocking it. It can't reach the active site. So right now, this arm, the thick filament, all it wants to do is grab onto that, but it can't because it's being blocked by tropomyosin. What controls tropomyosin? Something called the troponin complex, right here. That's holding it there. What you're going to see later on is when calcium levels go up in a cell, we talked about where those come from, remember the SER. When they come from the sarcoplasmic reticulum, they go up in the cell, that troponin complex is going to change its shape. And that change in its shape is going to move this rope to the side. And that rope moving to the side is going to expose the active sites, and now the myosin molecules can grab onto the rope and pull. And that's what's going to lead to muscle contraction. Okay? Again, take it piece by piece, you'll see it as we go on. Okay? So the troponin complex responds to calcium, moves the tropomyosin rope out of the way, exposes the active site of actin so that myosin can bind. So this is another way of looking at the same thing. Again, here are your heads of myosin or your thick filament, and they're waiting to grab onto that rope. Right now they can't grab onto it because the active sites are not exposed. So this muscle is not contracting. This muscle is stationary, resting. This just shows you all three of those together. This is another picture from another book. It just shows you the same kind of thing. I just show you this just so you can see it from a different point of view. The only thing I realized is when the other classes, it reverses the colors. So if it confuses you, you can skip over that. Okay? This is also another picture that shows the same kind of thing. Now, the only thing I'll show you here is this is a nice visual when you're looking at striations, what you're really seeing. Because you can't tell it very well, but actually right underneath this T-tubule is a Z-disc. That's how they're actually oriented. So you can actually imagine how that's put together. Now we talk about this being a triad, and that's not a term I'm ever going to ask you to know, triad. But all it really is it's this right here, this terminal cisternae, that's just that blue region of the sarcoplasmic reticulum, this blue region of the sarcoplasmic reticulum, and the T-tubule all put together is a triad. Okay? That's all that means. All I'd ask you to know is the sarcoplasmic reticulum and the T-tubule. They'll be the two things you definitely need to know with respect to that. So let's look at this picture here, because I think this is just something that your book doesn't do very well. And that's to really show you what happens with a muscle contraction. So again, I apologize if the colors are reversed. So here, what I want you to pay attention to is the Z-disc right here. So here are the thin filaments, here are the thick. Once I contract the muscle, once I give the stimulus for contraction, which is an increase in calcium, these guys are going to grab onto the rope and they're going to pull. Now look what happens. See how the ropes get pulled in? See how the ropes get pulled in? And the overall effect is that the Z-disc gets, every Z-disc gets closer. So imagine going from this to this. That's what's happening. Does that make sense? Again, sarcomere, the smallest contractile unit of a muscle. If the sarcomere gets shorter, the entire muscle gets shorter. Why? Okay. Think about a train, right? What is a train? It's got a bunch of different cars, all connected end to end to end. That's exactly what the sarcomeres are. If you look at a muscle, it's sarcomere, 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 over and over and over again. If you make one train car get shorter, or make all the train cars get shorter, guess what happens? The entire train gets shorter. The entire muscle gets shorter. That's what happens here. So the distance between the Z-discs gets shorter on every single sarcomere in the entire muscle, and as a result, the muscle gets shorter. All at the same time. That's how this occurs. How does it all occur at the same time? Again, goes back to this idea. This amazing machinery right here called the sarcoplasmic reticulum. And this amazing, basically conductive pathway, so that as soon as that electrical impulse comes down the muscle cell, it causes a rapid contraction by releasing calcium. It's all triggered the same. So if you look, it wraps around everything in the entire cell. So boom, instant contraction. You okay so far? We're getting a little more complicated, but
Can you go back to the other slide? Can you say the sarcoplasmic reticulum? Are you talking about the whole thing?
sarcoplasmic reticulum is the blue.
Okay.
Everything you see here in the darker blue, not the light blue.
Okay.
Darker blue, yeah. So we're going to start here on the middle of page 8. Physiology of a skeletal muscle fiber. We're going to talk about something called an action potential. Okay? An action potential. We haven't talked about that quite a bit. We introduced it very briefly in chapter 3. So if you remember, you have your plasma membrane, and this is true for a muscle cell as well. It is slightly negative on the inside and slightly positive on the outside. And we maintain that all the time. We have the sodium-potassium pump. So it runs all the time. It's like a sump pump in a house. It always runs to make sure your basement stays dry. So we are constantly pumping out sodium, and we are constantly pumping in potassium, so that we have a gradient built up of 10 times sodium and 10 times potassium. But due to the leakiness of channels, because these guys are going to, the potassium's going to constantly come back, the sodium's going to constantly come back. We're constantly keeping the pump on though, but due to that leakiness, we end up with a slightly negative inside membrane. Remember all that? Hopefully. So as a result of that, we end up with about negative 70 millivolts. That's the charge on this membrane. That's the resting membrane potential. So in a cell that's not stimulated, it's just sitting there normally, we maintain negative 70 millivolts. What we're going to see later on is, let's say, voltage here. Okay? And this would be negative 70. So there's going to be a region of our plasma membrane that starts off at 70, but we're going to see this rapid increase, and then we're going to see a rapid decrease in the voltage, going up to roughly positive 30. And then we're going to see it coming back down again, going actually below this. We won't talk too much about that today. And then coming back to resting membrane potential about negative 70. So this actually occurs in one area of the plasma membrane. So you can imagine if this is a large, here we go. So this right here is a muscle fiber, muscle cell. So right here, at this particular point, we all of a sudden have a spike. So the voltage goes from negative 70 up to 30, and it comes back down again. Then it travels over here. Here, 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 here. So we have this large spike that then starts up, comes back down, and goes back to resting, and that propagates all the way down the entire surface of the cell. Does that make sense in general? That change in voltage is short-lived, but that it spreads all the way down the cell, and that is a voltage, or a change, or a current, as we call it. So we're going to be talking about how this voltage changes going down the plasma membrane. And that's actually what goes down the T-tubules, and that's actually what triggers calcium release. You'll see more about it. I'm kind of jumping ahead here. Just didn't want to throw you later on. Might not give you just a bit of an introduction. So that's what we call an action potential, okay? The electrical current. Let's talk about what happens at the muscle. So let's say that we want to move a muscle, right, voluntarily. This is a skeletal muscle after all. So we have some sort of impulse, or we call it action potential, that comes down in a neuron. Now, the part of the neuron we're going to be talking about is called an axon. If you know what that is, great. If you don't worry about it at this point. But it's a long tail of a neuron. And these guys are going